Good evening, everyone. My name is Jane Pickering, and I'm the Executive Director of the Harvard Museums of Science and Culture, which includes the two museums that are sponsoring this evening's event, the Peabody Museum of Archaeology and Ethnology and the Harvard Museum of Natural History. I'd also like to give a warm welcome to our virtual participants because we are live streaming this lecture onto Facebook, so welcome to you as well. And we're delighted this evening to have Athena Aktipis, just been practicing that, from Arizona State University with us tonight, and she's going to discuss her research on how cancer is intricately linked to the evolution of multicellularity. Before we get going on that, though, I would just like to mention that you can find out about all the programs sponsored by HMSC in the program guide, and you can find that to the table to my right, and you can also, at that table, find out how to join our email lists and also how to become a member of HMSC and help support programs like the one this evening. The guide is also available online at hmsc.harvard.edu. So we have many exciting events. This is one of many this semester. So I'll just mention three, actually, before we move on. This Thursday, so tomorrow, the Harvard Semitic Museum will present a lecture on how digital technologies are being used to understand the construction of the great Egyptian pyramids. Then on Tuesday, April 10th, the Peabody Museum will present a lecture on the archaeology of the Great Aztec Temple found in downtown Mexico City. And then on Thursday, April 12th, the Harvard Museum of Natural History will present a lecture on how artistic photography can be used to understand the natural world. So we have many great things coming up, but also a marvelous thing coming this evening. And it is now my pleasure to introduce Barbara Natterson-Horowitz, Professor of Medicine at UCLA and visiting professor in Harvard's Department of Human Evolutionary Biology, and she is going to introduce our speaker. Thank you.
It is a real privilege and pleasure to introduce our speaker, Dr. Athena Aktipis. Dr. Aktipis is an evolutionary biologist, an ecologist, and a cooperation theorist who applies models that are usually used to understand how plants and animals relate to one another in an ecosystem to human areas that aren't traditionally looked at in that way. Her work considers the common elements of cooperation between individual humans living in a group, the body's cells living together in an organ, and even cancer cells side by side in a tumor. And I believe you're going to be hearing more about that tonight. Dr. Aktipis received her bachelor's from Reed College and her PhD from University of Pennsylvania. She is assistant professor in the psychology department at Arizona State University, the co-director of the Human Generosity Project, and co-founder of the Center for Evolution and Cancer at the University of California at San Francisco. She's the author of a forthcoming book to be published this March, Evolution in the Flesh, Cancer and the Transformation of Life. On a personal note, I'm a physician, and several years ago I started a program inviting evolutionary biologists to lecture to and join our physicians, our interns, and our residents and our medical students on rounds at UCLA. The idea was to expose physicians to evolutionary concepts and encourage them to incorporate these ideas into how they're thinking about human medical problems. Well, Dr. Aktipis was one of the first evolutionary biologists to participate. And her presentation, the concepts and the models, disruptive as they are to traditional, conventional ways of thinking about cancer, sparked tremendous and fortunately ongoing excitement about evolution and medicine at UCLA. So, Athena, thank you for making our program look so good. Tonight you are in for an intellectually exciting experience, and I can say that because that is exactly what it has been for me each and every time I've heard her speak. Dr. Aktipis.
Thank you so much for that wonderful introduction, and thank you to all of you for being here and to Harvard Museums for having me. It's been a really wonderful trip so far, and I'm really looking forward to the opportunity to speak to you all tonight about my work on cancer and this question of why cancer is so prevalent in life, not just among humans, but across life. So I want to start with one of my favorite pictures. This is a crested cactus, and this picture is one of the reasons that I started looking at cancer from an evolutionary perspective. My work up until about 10 years ago was really focused on the evolution of cooperation. When does cooperation evolve? What makes it stable? And when can it break down? But one of the things that really drew me into looking across life was seeing specimens like this that are quite rare, and they have these features that seem cancer-like in some ways. But plants like the saguaro are actually living with this kind of cancer-like growth. And I was fascinated by it, and fascinated by the question of, is this cancer? And what does cancer look like across life? And fundamentally, how can an evolutionary approach and a cooperation theory approach help us understand this? And just as a side note, I became interested in these cacti long before I moved to Arizona, where we actually have some of these specimens. And I admit that maybe they were part of the draw for moving to Arizona. You actually sometimes see them on the side of the road or when you're hiking, and they have this sort of sculptural beauty that is really amazing. So, the question is, how do these plants live with cancer or live with these abnormal growths, whether we want to call them cancer or not? So this is actually part of a more general phenomenon called fasciation, where plants will have their the meristem, which is the tip where the sort of plant stem cells are, rather than it being a few cells, it becomes a band of cells that are proliferating. And then it creates this sort of sometimes like a brain-like pattern, like this specimen below, or this sort of cresting, or sometimes plants that almost look like they're something out of a Dr. Seuss book, because of the way that the meristems are growing in this band rather than in a point. And you actually see this across many species of plants, so it's not unique to cacti. And so what I want to talk about today is how cancer really is this fundamental component of multicellular life. And we need to start at the beginning of the origins of multicellularity, because that's where the story of cancer starts. And when we do that, we see that cancer is actually a normal part of multicellular life. And in fact, that cancer is, in a way, a sort of cellular tragedy of the commons. That it's a situation where cells are exploiting normal cells, and that's having a cost on the whole organism. So it's fundamentally a dilemma of cooperation that's happening on a cellular level. And what taking this broader perspective has shown to me, and I think moved our understanding of cancer forward to realizing that cancer is not necessarily something that we need to treat extremely aggressively in all cases, but that cancer may be something that we can live with, and we can use an evolutionary understanding to better treat and prevent cancer. And at the end of my talk, I'll come back to some of those implications for treatment. So let's start with a very basic question. What is cancer? Well, it turns out that there are a lot of approaches to answering this question. There are a lot of different definitions of cancer. In the clinic, oftentimes, invasion and metastasis are the deciding factors for if you're going to call a tumor cancerous or not. Pathologists sometimes will point to having dysregulated differentiation, meaning the cells don't look like they should for the tissue that they're in. They're in a different state than what they should be in. And then there's the hallmarks of cancer approach, which is has basically looked across human cancers at what is common across cancers in terms of the kinds of characteristics you see in cancer cells. And then today I'll talk about the approach of looking at cancer as cellular cheating and how that can help to unify some of our concepts of cancer. So the approach in the clinic and the definition in the clinic is focused on when does a tumor invade into the bloodstream and create new growths. And this is a process that actually has a lot of similarities to processes in ecology where organisms evolve to move and colonize new environments. So I'll come back to talking about that in the context of a model that I've done of the evolution of cooperation a little bit later. And then this is the hallmarks of cancer approach. So this is based on a really excellent synthesis of the literature done by researchers Hanahan and Weinberg, who looked across cancers and found that these characteristics appear again and again across cancers. So we can focus on invasion and metastasis, or we can look at these hallmarks, or pathologists will focus on often the dysregulated differentiation. So all these different approaches. But the other approach is to think about cancer as a similar problem that we face anytime we have to scale up cooperation. So I study cooperation. That's my main topic. And I'm equally interested in cells and how they operate as a unit in order to cooperate and make our bodies function. So if you stop for a moment and think about it, we have approximately 30 trillion cells in our bodies that are cooperating with each other, coordinating their gene expression, coordinating on a microsecond level in order to make us able to breathe and eat and sit here and think and listen to the talk. So it's just really, when you stop to think of it, totally astounding, the level of cooperation that's happening in our bodies. And the level of cooperation that happens in our bodies is far beyond any level of cooperation that we could achieve as a human society in terms of the kinds of things that our body accomplishes cooperatively. But every time you scale up cooperation from very small scale interactions to large scale interactions, you have opportunities that come from that, right? So you can create more public goods that you can share. You can sometimes manage risk better. There are opportunities from scaling up. But the larger your group is, the bigger the problem of cheating is, and the harder it is to find individuals who cheat and keep them from destroying the group. And so no matter what kind of group you're talking about, whether it's a group of cells or a group of humans, as the group size gets bigger, the opportunities go up, but the problem of detecting and suppressing cheating becomes larger and larger. And so if we look across organisms, and in my field of cooperation theory, a lot of different solutions have been proposed to this problem of cheating. I won't talk about all of these, but they roughly fall into 3 categories. One category is essentially you have some sort of tit-for-tat mechanism, reciprocity, or you're choosing partners. Another is that there's you get individuals that are genetically very similar, and so that allows cooperation to take off. And then there's another set of explanations around detecting cheating and suppressing cheating. And when it comes to understanding cancer, we have sort of the question of, well, which of these solutions, which of these classes of solutions are getting used the most? And one that I'll focus on today is this idea of cheater detection. And this is some work that my graduate student, Andres Munoz, is doing. And we're working together to look at, in humans, how do we actually detect violations of social rules? Like, for example, you should only ask for help if you're really in need. And we find that people are much better at detecting violations of rules that have to do with social interactions than they are at detecting violations of arbitrary rules. So in humans, we know cheater detection is something that we do, and it's probably an important part of maintaining cooperation in large-scale societies. So might there be some of these similar principles happening in cellular societies? Well, what I'm going to talk about today is how some of the mechanisms that we have thought about as cancer suppression mechanisms really come down to detecting and suppressing cellular cheating. And that in order to make a cellular society, and here I'm thinking of all kinds of cellular societies, from small groups of cells in early multicellularity to large groups of cells like our bodies. But in order to have those cellular societies be viable, we need some mechanism for finding and detecting cells that are not actually behaving in accord with those rules. And so the idea of cellular cheating applies to many different issues in cancer. I'll talk today about the cancer hallmarks, about invasion and metastasis, about how it can help us understand cooperation or cancer across species, and the evolution of cancer suppression systems. So this idea of multicellular cheating, I think, can help unify a lot of these ways that we're looking at cancer. But in order to really answer this question of what is cancer, then we have to go back, all the way back to the origins of multicellularity, and understand, well, why did multicellularity evolve in the first place? And there are benefits from cells clustering together so they can get protection from predators. They also can produce things that are good for their neighbors, and by producing it together, they can get some benefit. And in general, as you get more and more complex, there's more opportunities to divide labor, have cells that have different cell types, so you can get more done and more different things done at all times. So it can be a benefit to scale up to be a larger organism instead of just a single cell. But this actually required cells to evolve capacities to cooperate and suppress some of what would be sort of natural behavior for the cells. So they evolved to inhibit proliferation, to regulate cell death, to transport resources around. Because once you get to a certain size, you can't rely on diffusion from the outside to get resources. So you actually have to have some systems for transporting resources to the cells that are on the inside. You have division of labor. And then you also have the creation and maintenance of the extracellular environment. So this is the environment that's between the cells. And there are all sorts of proteins and factors and things in between the cells that hold the cells together and that provide a sort of supportive matrix for the cells. But it's easy to trash that as a cell if you're not in the right gene expression state, if you're using the resources that are in your environment too much, or if you're creating a lot of waste product. So all of these things needed to evolve for multicellularity to be viable. And if we look at, well, what does cancer look like in terms of these foundations of multicellular cooperation? we see that cancer actually cheats in every one of these sort of cooperative rules that make multicellularity viable. So cancer cells will proliferate without limit. They actually have mechanisms for avoiding cell death. They'll monopolize resources. They will not sort of do the jobs that they're supposed to do for the tissue that they're in. And sometimes they'll co-opt the labor of other cells that are nearby to benefit them. And they will destroy the environment around them often. And so you might at this point be wondering, well, how am I defining cheating? And is this really cheating? Don't you need sort of an intention to cheat to call it cheating? Well, the way that I define cheating is sort of very operational, like what is the actual effect here that's happening? And whenever you have a group that has some shared rules, and those rules could be explicit, like, say we all agree that we're not going to exploit a common resource. That could be an explicit rule. Or it could be implicit, like if cells have genes that regulate their use of resources. So that's a rule that's shared, but it's implicit, and it's based in the genes that are shared among those cells. And so when the breaking of that shared rule leads to an advantage for the individual violating the rule, then we can call that cheating. And in this sense, cheating, cellular cheating, isn't a metaphor. It's just what's happening on the cellular level. You have cells that are taking advantage of the cells around them that are following the rules, essentially. So in this sense, we can think of multicellularity as a sort of massive public goods game where all of the cells in that multicellular body are behaving in a way that's good for the multicellular body, but then some cells are cheating and getting a benefit from that, a temporary benefit, right? Because they're not, with some exceptions, cancer is not actually transmissible, so there's a limit to how long those cancer cells can have an advantage, but still they're getting this temporary evolutionary advantage. And in general, one of the reasons why it's possible for our bodies to have very high levels of cooperation is because we're largely made of cells that are genetically identical, that have all of these rules encoded for cellular behavior. So this genetic relatedness among cells is one of the reasons why we can have this massive, large-scale cooperation. But even within our bodies, there are a huge number of cells that are mutated. In fact, the level of mutations, even in normal tissue, is really quite high. So we're largely made of cells that are genetically identical, but not entirely. And anytime you get a mutation, it's possible that that mutation will be in genes that are responsible for cellular cooperation or for the mechanisms that help to detect cellular cheating. And so when you get these mutations, they have the potential to begin on that pathway to cancer. And so how is it that cells actually cheat? Well, they can signal for resources. So when they don't actually need resources in order to sort of do their proper jobs, they can proliferate when they shouldn't. They can fail to die when they should. They can shirk their cellular duties in the division of labor sense. And they can exploit the cellular common. So there's a lot of routes to cellular cheating that are possible. And then if the mechanisms for detecting cellular cheating aren't in place, you get a situation where these cells, they grow in frequency, they can accumulate even more mutations, which can initiate cancer or accelerate cancer progression. So that's how you go from a sort of cooperative multicellular body to a body that has cancer or cancer-like phenomena in it is through this process where cells that are sort of cheating end up getting an evolutionary advantage over cells that are behaving in a sort of proper way for that multicellular body, that don't have mutations in the systems that regulate cooperation and detection of cellular cheating. Excuse me. So this is one way that we can sort of visualize what's necessary for the evolution of multicellularity. We have the inhibition of cell proliferation, control of cell death, and these two parts up here, they really control the sort of population size of the tumor. As if the cells aren't dividing or they're dying when they should, then that regulates the population size. And then on the bottom here, we have resource allocation, so having some way of getting resources where they need to go, division of labor, and extracellular environment maintenance. And these three on the bottom, they are sort of more ecological, almost economic, you could say. They have to do with how a multicellular body is using resources, using the environment, and creating the environment, and what kind of work is really being done by the cells. So these are really the foundations of multicellular cooperation. And in cancer, all of these foundations break down, as we've sort of talked about before. Now, one thing though, when we start putting all of these foundations into this framework, mapping how cancer breaks down, then we can see, well, how do these features map on to the hallmarks of cancer that I talked about earlier, that Hanahan and Weinberg identified, these features that are similar across human cancers. So me and a group of other cancer evolution scholars worked together to try to map the hallmarks of cancer, which are here on the outside, with the foundations of multicellular cooperation, which you see on the very inside. And amazingly, the hallmarks of cancer map on very well to these foundations of multicellular cooperation. So on the top, 5 of the hallmarks really have to do with sort of the regulation of the size of the organism. And then on the bottom, there are a few hallmarks that have to do with sort of monopolizing resources, using resources too much. And then on the bottom here, we have dysregulated differentiation, which, I don't know if you remember, it's not something that was in the hallmarks. So it's sort of, this framework suggests, well, maybe this is a missing hallmark. But it's also the way that pathologists often identify cancer. So this seems to really help to fill a gap of what might be a missing hallmark of cancer that emerges sort of from these first principles about the evolution of cancer and the evolution of multicellularity. And then over here, invasion, I mentioned before, is clinically the important thing that is focused on in terms of whether something should be defined as cancer or not. And it doesn't neatly fit into this environmental degradation, but having environmental degradation is something that makes it easier for this invasion to happen. And here, the activating invasion, we suspect that this might actually be a downstream consequence of many of these other processes that are happening. I'll have a chance, I hope, to talk to you about that a little bit later in the talk. So returning to sort of the big picture, it seems like cancer is this fundamental part of being multicellular. And if that is the case, right, then it's something that, in a sense, is an unavoidable consequence of our, very, very ancient ancestors deciding, so to speak, to be multicellular. So maybe we could be cancer-free, but then we would be single-celled organisms. So maybe that's not such a good deal. So back to these crested cacti. So I got obsessed with these almost a decade ago, and then that led me into thinking about cancer across life. And I had the pleasure and honor to be part of a one-year working group on the topic of cancer evolution at the Wissenschaftskolleg in Berlin. It's an institute for advanced study. And we had the chance to spend a whole year thinking about and talking about what are the important open questions in cancer evolution. And one of the biggest questions that we all had was, what does cancer look like across the tree of life? And so we spent the better part of the year reviewing this literature, just looking at, are there reports of cancer in species across the tree of life, and focusing in particular on complex multicellular organisms. So the Wissenschaftskolleg is a wonderful place to do this because well, in a way, it's sort of like summer camp, but for professors. They feed you lunch every day and breakfast if you don't care to make your own breakfast. And then you can just sit in a room with a bunch of other really amazing, creative, smart people and work on questions. So it was a great opportunity, and I also had the chance to bring along my postdoc, Amy Body, who you'll hear about later. And so we worked together on this question of what does cancer look like across the tree of life? So this is the result of almost a year of work. Let me orient you a little bit to this figure. It's a phylogeny. So what this means is that each of these taxa evolved from a common ancestor back there, and then you can see sort of how far they are apart in evolutionary terms from looking at when they diverged. So here, these boxes, they tell you if the form of life is characterized by really complex multicellularity. Those are the black boxes. More simple multicellularity, those are the gray boxes, or unicellularity, those are the white boxes. And then next to them, you'll see red, yellow, or green squares, and those show you whether in our literature review we found reports of cancer. Those are in red. Whether we found cancer-like phenomena, so those are dysregulated differentiation or abnormal cell proliferation, but something that doesn't actually have invasion or metastasis. And then the green are taxa for which we found no reports of cancer. Now that doesn't mean they have no cancer, it just means that in the literature that we looked at, there weren't any reports yet. So we can start up here with animals and look a little bit deeper into into the tree of life here. And what we see is that really cancer is observed across almost all the taxa invertebrates, in mollusks. This is a picture up here from a starfish, and that arrow is pointing to melanocytes. So that's basically a starfish melanoma, a bunch of melanoma cells. And down here we have slides from a clam. This is a blood cancer, essentially, in a clam. So we see cancer across animals. We can look at some of the sort of simplest, earliest forms of multicellularity, cnidaria. And there we see both cancer-like phenomena and cancer itself. So in coral, there's a cancer-like phenomenon where you get a loss of proper differentiation. And in hydra, you see growths that look like tumors that much larger and more complex forms of multicellularity have. In fungi, so multicellularity actually evolved multiple times independently in fungi, and there are many different types of abnormal differentiation that are observed in fungi. And this is just one example where you can see that the patterns of cellular differentiation that should be happening on the bottom of the mushroom are actually happening on the top. And perhaps my favorite, we have our green algae and plants here, and this is where we see this wonderful fasciation, this cresting phenomenon, where the meristem sort of expands into a band. Surprisingly, we also found a report from 1945 of what looked like a metastatic growth in a plant that wasn't associated with bacteria. But that was the only piece or only report that we were able to find of something that looked really like cancer in the sort of clinical sense of the word. So this is another way of looking at the evolution of life and its relation to cancer suppression. So we have here on the bottom indication of the stages from the evolution of multicellularity, and it's sort of hard to see here, but in this box, this is when metazoans evolved. So this is when we started to get the kind of multicellularity that we have today. And in that phase of our evolutionary history as multicellular organisms, we see an overrepresentation of genes evolving during that time that are responsible for both the cooperation among cells. So those are what are often called oncogenes, because if they break down, they contribute to cancer. So those cells that are involved in cellular cooperation. And we also see an overrepresentation of genes that are involved in tumor suppression. Those are sort of the cheater detection genes. So multicellularity required both the cooperation of cells and these mechanisms for regulating that cooperation and enforcing that cooperation. And so this, for me, when I realized this, it started to shift how I was thinking about cancer and about multicellularity. And I realized that you can think of this transition from unicellularity to multicellularity as, in a way, what was required is that life solve this problem of cellular cheating. And if that's the case, then essentially cancer or cancer-like phenomena are part of what might have kept life unicellular for so long in the history of life. And so life itself basically had to evolve to suppress this cellular cheating. And one way of doing this, sort of ironically, is for life, in the sense of multicellular life, to actually evolve to suppress evolution happening inside the body. And that is one way that you can actually restrict the evolution of cellular cheating, because you're keeping those cells that have an evolutionary advantage because of cheating, you're keeping them under control. So when we start looking at the evolution of larger body size and longer lifespan, this problem of the evolution of cheating within becomes larger because there's more opportunities for cellular cheating, and then this actually selects for even more suppression of cellular cheating. And this is the reason for what's called Peto's paradox. So Peto's paradox is this finding that cancer susceptibility is actually doesn't scale with body size. Now, you would expect if the chance of getting cancer is just a matter of how many cells you have and how many times they divide, that larger organisms should get more cancer than smaller ones. But they don't, because these larger organisms also have better mechanisms for suppressing cancer because they've been under selection for that. And this is a graph that shows cancer rates among mammals. And on the bottom, we have the mass. So this is body size times lifespan. And we see larger size, longer lifespan, and then the number or the proportion of the necropsies that have tumors in them. And here, among mammals, there's really no relationship. If anything, it's maybe slightly negative, that larger organisms get less cancer. We're now undertaking a very large project led by my former postdoc, Amy Boddy, who was with us at the Wissenschaftskolleg working on this review of cancer across life. And she has been working tirelessly since she came to work with me many years ago to build relationships with zoos and with veterinary pathologists to get access to data on the prevalence of neoplasias and cancers across many, many species. And she has 77 species of mammals, 20 species of reptiles, 39 species of birds, and 2 species of amphibians. And altogether, a huge number of individuals in this database. More than 15,000 of them have abnormal proliferation, and the overall prevalence of these neoplasias or these abnormal proliferations is 18%. Now, this is early data, and it's unpublished, and it's a work in progress, but I wanted to show you what we're sort of finding at the moment, which is that so across animals, like I said, we have about an 18% prevalence. Amphibians, it seems to be quite low. Birds, relatively low. Mammals, like us, 26%. That's quite high. And humans, maybe slightly higher than that, but not that much. And reptiles, somewhat low. You might be interested to know the mouse lemur, 65% of the necropsies had abnormal proliferation, and alligators seem to have the lowest risk. So I don't know if, I don't know why, and I don't know that I would necessarily want to be an alligator, but they seem to get very low rates of cancer. And if you look overall, there's a significant effect of body size and lifespan here, that the larger and longer lived you are, the less cancer you get. So again, showing Peto's paradox, that larger body size, longer lifespan is not actually correlated with cancer risk, probably because there are more mechanisms actually for suppressing cancer. So now if we think about what's actually happening within the body, cancer is evolving, or cancer cells are evolving to exploit the resources of the body, to exploit the ecology of the body. So I'll show you an example of this, which is the way that cell resource use affects the evolution of cell movement. So we've talked a little bit about this idea of the tragedy of the commons, but basically, cancer is essentially a tragedy of the commons on the cellular scale. So this sort of problem of overgrazing. Every shepherd has something to gain from having their sheep overgraze on the commons, but everyone does better if everyone holds back a little bit. And in the context of a body, cells are constantly needing resources and using resources, but normal cells sort of hold back in their resource use and don't use everything that's around. But cancer cells overuse resources. They have upregulated metabolism, and their energetics are totally dysregulated often. So I created a model of this. This is a cover image from a paper that we published in Cancer Prevention Research. And this is a spatial agent-based model where cells are sort of living in this world where there are resources coming in to every one of these little patches that they can exist on. And they consume those resources. And cancer cells, the only difference between the sort of cancer cells and normal cells in this model is that the cancer cells are consuming higher resources. And then what I looked at is how does the rate of cell movement change? How does it evolve if I let that parameter evolve? And so I made 10 runs where I looked over many time periods to see what happens when you have these differences in the metabolic rate, the rate at which cells are using resources. And what I found was when the metabolic rate was high, so this is when cells are using resources much more quickly, that they actually evolved to be much more mobile. So here we have cellular cheating in the form of overusing resources, actually leading to the evolution of cell movement, which we know is an important part of invasion and metastasis. And in general, resource monopolization may be an important driver of metastasis because we know even that normal cells can respond to low oxygen by leaving an environment. So, and this prompted me to ask a sort of follow-up question, which is whether this resource use can not just lead to higher rates of movement, but can make cells sort of more ready to move if the resource levels go down. And this is unpublished data, but what I'm finding in my preliminary investigations here is that higher resource use actually makes these cells evolve to leave more readily. So we have conditional movement happening on the part of cells when they have these high rates of resource use. And what this means, and now I'm kind of going a little bit into some of the work I've done on cooperation theory and what conditions allow cooperation to evolve. But what this means is that you can have these dynamics where cells actually will be more likely to stay in better tumor microenvironments and leave in worse tumor microenvironments, which might seem like it's a good thing, but this kind of dynamic actually can select for cooperation among cancer cells, which we do not want. I think this is one of the really interesting open questions. How do cancer cells evolve cooperation? And these kinds of dynamics of interacting with resources and the local environment, I think, will be a really important piece of how we will understand what's actually happening during this transition to metastasis and when cells evolve to cooperate with each other at the expense of the multicellular body. And further, in order to really understand what's happening during this, cancer hallmark of invasion and metastasis, I think we have to actually take into account how cooperation is breaking down in many of these domains of multicellular cooperation. So in a way, understanding metastasis and invasion, I think, requires us to take all of these different aspects of cellular cheating and think of them together and what kind of feedback they give to each other in order to make those conditions more likely for invasion and metastasis. So I've talked about how this idea of cellular cheating can help to unify a lot of our concepts of cancer. That cancer is sort of this manifestation of a fundamental tension between cooperation and cheating that is just part of the evolution of life. The cheating framework can help us understand the cancer hallmarks. It can help us understand when does invasion and metastasis happen. It can help us understand why cancer exists across life. And it can also help us understand the evolution of cancer suppression, because that cancer suppression is essentially cheater detection, cellular cheater detection. So now we can ask the question of how exactly would a multicellular body detect this cellular cheating? And it turns out that we actually have an amazing multi-layer system of cellular cheater detection. There are mechanisms that are intrinsic to the cell. You can sort of think of these like the cellular conscience. The cell is actually monitoring itself for DNA damage, for proteins that aren't folded properly, for is it proliferating too much, anything that it's doing that's out of the ordinary. The cell actually has internal mechanisms for kind of keeping tabs on its own behavior. There are also neighborhood mechanisms. So cells will monitor the cells that are near them. Are they adhering properly to the cells in the environment? Is the tissue architecture normal? Are there cells near them that are surviving when they shouldn't? And then on top of that, we have the system-wide mechanisms. And those, the best example is our immune system. And our immune system is actually constantly sort of monitoring the body for regions where there might be abnormal proliferation, too much resource use, waste production, and then allocating more immune cells and more immune resources to those regions. So we have all of these systems for detecting and responding to cellular cheating. And part of what multicellular bodies do is essentially try to regulate this cheating so that we end up with something that looks more like that middle, as opposed to these things on the edges that can be contributing to cancer. So one mechanism that we can look at is a gene called TP53. It produces a protein called P53 that in some ways we can think of as maybe not the cheater detector of the genome, but a cheater detector in the genome. And what P53 or TP53 is, it's this central node in a really complex genetic network that's integrating signals from many diverse pathways from the cell, and then it decides, should the cell just go along with its business and complete its cell cycle and divide? Should it stop instead and repair DNA? Are there things that are broken that need to be fixed before it should replicate? Or is it so beyond repair that it should undergo programmed cell death, that it should just commit apoptosis and die so that it doesn't pose a risk to the organism? And so if we look at how P53 interacts, we see that it's actually a central node in this really large network, which seems kind of puzzling on first pass. Why would it be at the middle of this network where it's so vulnerable to being mutated? And in fact, if you get a mutation in P53, that can create a situation that predisposes you to cancer. So why would it be so vulnerable? Well, part of the answer might be that it needs to be integrating information from all of these different sources from the genetic network in order to sort of make the right decision of if a cell is problematic or not. So some of the pieces of information that can go into this decision, include the signaling that cells are doing for resources, the proliferation they're doing, sort of all of these things that tie into the foundations of cooperation. And if we look at the gene TP53, we see that large organisms like elephants actually have more copies of this gene than humans. So we have 2 copies. Elephants have 40 copies, and their cells are more likely to undergo this cell death when they're exposed to radiation. So it seems like they are upregulating those cancer suppression mechanisms in order to, in a way, deal with having such a large body size. So cheater detection, cellular cheater detection, seems like it's actually a really, really important part of making complex multicellularity large, long-lived organisms viable. And having it allows these organisms to have a reduced likelihood of cancer-related death, which actually means that these cancer suppression mechanisms may have really facilitated the evolution of large body size and long lifespan. And there's some evidence of this actually in elephants that these copies of TP53 actually evolved along with increasing body size. And the body size went up, and then the copies of TP53 kind of got selected afterwards. So what all of this means is that, in a way, our multicellular bodies, with their 30 trillion cells all processing information and responding to the cells around them and cooperating and coordinating, in a way, have a kind of collective cellular intelligence that is detecting and responding to cellular cheating in order to keep us maybe not cancer-free, but to keep cancer rates lower than they would otherwise be, and to protect us from those threats of cellular cheating. And if we do have cancer-like mutations, potentially keeping those under control so that they don't have such a negative effect on our survival. So we've just talked about the history of cancer. Well, what does all of this mean for the future of cancer? So will we be able to actually completely eliminate cancer? Probably not, because it's a fundamental part of multicellularity. But evolutionarily informed treatments may help us do a better job of controlling cancer. So if we can support the cooperation of normal cells in the body, support the cheater detection systems that we have in the body, like genes like P53, if we can disrupt the cooperation of cancer cells before they get the kind of abilities that really threaten us, we can potentially keep these growths under control before they really become a clinical issue. If we can slow down evolution happening in the body, that's also one route to controlling cancer, and minimizing the evolution of resistance, because really resistance in the clinic is what kills. Because when a tumor stops responding to a drug, that's because the cells have actually evolved resistance. And so we can look at a few of these. One that is particularly interesting and apropos to our previous conversation is there's work right now looking at whether can actually translate these findings from elephant TP53 into the clinic for cancer suppression. And then there, in terms of slowing down evolution, there is evidence that using non-steroidal anti-inflammatory drugs can actually reduce the mutation rate by about an order of magnitude and reduce the risk of progression from a premalignant condition called Barrett's esophagus to esophageal cancer. And so to the extent that we can actually reduce the mutation rate, then you can get an extension of lifespan by quite a long time, potentially, because you're reducing the rate at which those cells are acquiring additional mutations that move them along on the pathway to cancer. Perhaps the most exciting thing in cancer treatment right now, in my opinion, is the approach of adaptive therapy. So we've known about this idea of adaptive therapy for about a decade. Was started by Robert Gatenby, who is a radiation oncologist at Moffitt Cancer Center in Tampa, Florida. And what he found was that he could actually control tumors in mice, in ovarian models and ovarian cancer models and in breast cancer models by conditionally giving therapy only when the tumor was growing and otherwise leaving it alone. And what this means is that you can start with a tumor here that has mostly cells that are sensitive to your treatment. So those are in purple, but maybe has a cell that is resistant to treatment. And if you use standard therapy, where you're giving high doses, you select for that resistant cell, and then you can no longer keep it under control with your therapy because all the cells are resistant. But in adaptive therapy, you give an initial dose to knock the tumor back so that it's not so large, and then you only dose when the tumor is growing. And what this means is that over time, you actually maintain a population of cells that are sensitive to your drug, meaning that they will respond by dying when you use your drug. And if you want to have long-term control of a tumor, then you want a drug to keep working, right? You don't want all of the cells to be resistant. And so this is currently in clinical trials, and just recently some data came out from their clinical trial. So this is in prostate cancer, which is the most advanced prostate cancers that have not responded to any other treatments. And on the top, we have the trials. So these are the individuals who are on adaptive therapy. And on the bottom, we have the controls. So these are the individuals who are not on adaptive therapy, who are getting the normal standard of care. And the black X's show where they had scans that indicated that their cancer progressed. And the triangles show when they had PSA measurements that indicate their cancer progressed. And you can see that for the controls who are not in this clinical trial, they had progression on, most of them progressed to full-on cancer that is untreatable. And so, but up here with the trial, very few of the individuals actually progressed based on the scan and based on the PSA measurements. So this is really promising. I mean, not even half of the patients have progressed yet. So we have no idea how well it's going to work, but it seems to be working really well. So I think this is a really inspiring and intriguing approach to cancer. And in a lot of ways, one that I think is consistent with what we know about cancer, which is that cancer is something that we have lived with as multicellular organisms since multicellularity began. And so if we can look to a future which is not necessarily about curing cancer and making cancer completely go away, but rather a future that is about how to best control cancer or cancer-like growths so that we can live with them and maybe die with them, but not of them, right? There's a big difference there. And so if we can keep our bodies healthy and keep our neoplastic growths under control long enough, we may be able to live very long and fulfilling lives, even with these growths. Multicellular life has coexisted with cancer since the very beginning, and so this, I think, is just an extension of what we know about the evolution of life, and we've evolved actually to suppress cancer. So let's bring it all full circle. These crested cacti, they have cancer-like growths, and they live with them, right? So when we're thinking about cancer and cancer-like phenomena, neoplasias, these specimens, I think, might have something to teach us. And in fact, when I was in Berlin at the Wissenschaftskolleg, I met actually the son of one of my colleagues there who's a master gardener in London. And we started talking about these specimens, because as I told you, I'm completely obsessed with crested cacti. So I'll talk to anybody about them who will listen. And we started brainstorming about actually creating a garden based on these amazing specimens. Now, over the last few years, this idea has grown and kind of metastasized now to ASU, where I am. And we're now working with what has become a huge team of artists, landscape architects, gardeners, designers. This is just a subset of our team sitting around a table, sorting out how we're going to create this garden. And it is really amazing when you work with a team on a project like this, you realize just how prevalent cancer is, because many of the people on our team have been personally affected by cancer. And because what we're working on is a garden about cancer, people are telling their stories about cancer and how this garden for them is a priority because they see how it is a way of sort of bringing to life and bringing out into the open how cancer is this thing that we share with all forms of multicellularity. And so here is our plan. It's currently being installed, like as I speak right now, which is extremely exciting. And it's quite large. Initially, our plan was a small garden, like about this size. But you can see how big it is because these are tables and chairs right here. Yeah. So we're so grateful for all of the time and effort that so many people have put into helping this be realized, and also the support of ASU in helping to take what initially was a seed grant for an arts and science project and help it grow into this garden. And just as a sort of side note, this phenomenon that we see in plants of fasciation, this sort of cancer-like growths, they're called fasciation, which means many facets. And one thing I've realized through working on this garden is that this is actually, I think, a really powerful metaphor for what happens to life when cancer comes into it, both on the sort of scientific and biological side, right? It opens up all of these complexities, but also personally and socially. When cancer comes into our lives, it makes our lives much more multifaceted and interesting. And I think that this garden is a way of talking about cancer and how to control cancer and how to live with cancer in a way that can help us see how cancer is really a part of us, but so is the ability to keep cancer under control, at least for long enough for us to have fulfilling lives. And I want to leave you with this wonderful quote from Charles Darwin, how he ended The Origin of Species. We got our name for our cactus garden from this quote. It's the endless forms, most beautiful garden. He says, "There is grandeur in this view of life. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved." And as someone who studies cancer, as well as the evolution of life in general, I can read Darwin's quote here and think, well, this doesn't just apply to thinking about how organisms evolve, but it also applies to thinking about how cells in organisms are constantly evolving and sometimes creating forms that are most beautiful and most wonderful, even though they can also be a challenge for the organisms that have them. And I think that this sort of frame of seeing how cancer is a part of life, how evolution happens even within our bodies over our lifetimes, is a wonderful and beautiful idea, and something that we as a species perhaps can embrace in order to understand how cancer is, it's not just something that can threaten us, but in a way it's also an intimate part of us, and our ability to keep that evolution under control and keep cancer under control is also a part of our capacities as multicellular organisms. And so with that, I want to thank my many collaborators and our funding institutions that help make this work possible. And again, thank the Harvard Museums, and thank all of you for making the time to come and hear me speak about cancer and the evolution of life. Thank you all very much.